So, you've seen the title, and of course, one of the big questions that humans continue to ask themselves is, where do we come from? What makes us human? What sets us apart? How did we get here? How did we get the way we are? We humans tend to live in very complex societies. We are able to talk to each other, and most of us consider themselves to be fairly intelligent. At least collectively, we have evolved culture, technology, and have, of course, had an amazing impact on this planet. And some of the facets, very basic facets of human social behavior, I want to highlight and then go on to ask, perhaps, what can we learn about or contribute to the answers to these questions by studying non-human primates. So, one important facet is that humans establish long-lasting bonds, very special among animals. Then, among mammals, we have male philopatry. This means that the men in the group, or the male animals in the group, they stay in the group, and the females disperse. But the classic mammal model is the other way around. So in the classic mammal model, females stay at home with their female offspring, so you have female-bonded societies, and males disperse. But in humans, it tend to be, and especially specifically in early humans, we assume that it was the other way around. So we have male philopatry, and then of course this raises the interesting question: which are the conditions that favor such a flip in philopatric behavior? And finally, one of the really most striking features is the absolute extensive amount of cooperation, generally, but specifically among males. And of course, we assume that perhaps it has evolved in the context of hunting and collective group defense. And as evolutionary biologists, what we do is we try to use the comparative approach. So by studying other closely or less closely related species, to understand what are the factors that give rise to specific traits in evolution. And of course, what comes to mind is are the apes, specifically chimps, and also bonobos, that we consider to be our closest living relatives, or they are our closest living relatives. So a great deal of research attention is devoted to these guys. There are two drawbacks, if you will. One is they're living in dense rainforests, and there are only two taxa, two species that we can compare ourselves with. Then there are also gorillas and orangutans, but it's sort of a sparse data set. So in addition to really shine light on the processes that give rise to these evolutionary dynamics, it's also worthwhile sort of to take a broader approach and also consider what other species are doing. And in this context, baboons have traditionally played a really large role. So when the first people like Jane Goodall and Diane Fossey went out to study great apes, at the same time Irvin DeVore and some other people from the US started the first systematic long-term studies of baboon behavior. And this was because baboons are typically savanna dwelling. They live in the habitat that we think early humans also evolved in. And baboons live in large and complex groups. So maybe they give us some insights into the factors that may lead to the evolution of cooperation, for instance, or male-female bonds. And of course, then there's also the idea that walking bipedally evolved in the savanna. Baboons are not very good at it, but they can do like 2 or 3 steps on 2 legs. So most of you will not know that there are, in fact, different species of baboons. Most of you will probably be familiar with the Hamadryas baboon. This is up here. They live in northeastern Africa and on the Arabian Peninsula, and they can be kept in small compounds like in zoos. So when you go to a zoo, most often they will have Hamadryas baboons. They have the ugly red butt and the nice white shiny coat. And you can see them sort of running around on some stones that they put in this enclosure. But most of the research really, so Hans Kummer was the pioneer in studying Hamadryas baboon, but really the long-term studies, they've all been done on the southern baboons here. So specifically the Chakma baboon that lives in southern Africa, including Botswana, Namibia, and South Africa, yellow baboons, and then the olive baboon. So these are the three most well-studied baboon species. The Kinder baboon has really only a few years ago come into our focus of attention. We know relatively little, but what we do know, it seems that they're fairly similar in terms of their social organization, that is, who ranges together with whom and when, and how they're organized socially, like the other savanna baboons. So collectively, I will refer to these four southern species as savanna baboons. And the savanna baboons, they conform to this classic mammalian pattern of female philopatry, female bondedness. So the females stay at home, and then you have the grandmother, and you have her daughter, and the sister, and her daughter, and female cousins. And so you have these extensive female networks, and typically these networks are arranged in a rank hierarchy. So you have the top matriline, so the top female animals, and then the second beta matriline, and so on. And then the males, depending on which species they're in, they disperse either when they're fully adult and able to conquer perhaps a new group, or when they're still relatively young. But this is, of course, to avoid incest. The Hamadryas baboons, they conform to a very different pattern. They live in a so-called multi-level society where you have at the very core a male with his females and their offspring. And several males that form these one-male units, team up in a clan. And then several clans team up to a band. And then when you go to a sleeping cliff in the evening, you may see over 700 baboons on that sleeping cliff, and collectively they form the troop. And what happens is that most of the Hamadryas baboons live in very sparse habitats. So they come together in the evening for shelter to form these groups, but during the daytime they split into ever smaller groups. Eventually you may even find just one male with his females in the search for food. And of course, in such societies, you have these really interesting dynamics and who groups with whom when they go out and forage or when they go out and search for water. So that's a very interesting topic. Unfortunately, they live in habitats where it's really hard to follow them. And so now, when I started 10 years ago now in Göttingen, my professorship, I thought, okay, I have studied Chakma baboons in Botswana. I lived 1.5 years in the bush, and now I'm ready to go back into the field. Where do I want to go? And we started looking at the literature, and I thought, this is funny. There's not a single publication on Guinea baboons. And then I found one when I entered the search term Guinea and baboon, and I found one publication on the friendship between a guinea pig and a baboon. So I thought, well, why is there such a dearth on information about the Guinea baboons? There were some studies in zoos, but like we knew nothing about what was going on in the wild. And then we eventually dug up one sort of ancient PhD thesis from 1981 that has never really made it into the proper scientific literature, and also some book chapters. And what we found was that the people who had in fact studied Guinea baboons in the wild had very different views of what was going on. So Martin Sharman, who had produced, it's actually a beautiful thesis, this beautiful thesis on the Guinea baboons in Senegal, he said they're living in a multi-male, multi-female group. And he studied 2 groups, or what he thought was 2 groups, for 1.5 years, and he produced very detailed data on the ranging behavior and feeding ecology, but he never was able to identify everybody. He really only was able to identify very, very few animals. And so therefore, basically the social structure remained enigmatic. And then Gilbert Böse, he studied them in the zoo in Chicago, and he said, no, it's not a multi-male, multi-female group like in the savanna baboons, it's much more like in the Hamadryas. So we really have 1 male units within groups. And then Galad Luong, they spent a few weeks in the park. They were also not able to identify individuals, but still they said, well, it looks like a multi-level system. And this is what we also thought when we started. So we thought, okay, there's little known. It would be very, very helpful to understand, to complete the baboon chart. So let's go out and establish a field site in Senegal. So Senegal is a beautiful country, very nice people. You land in Dakar, it's eternally 24 degrees centigrade and a nice breeze is blowing and lovely fish and everything. And then you get into a car and you drive for about 10 hours and then it's 48 degrees. And it's just so scorching heat that you have to work in. But nevertheless, there in the Niokolo-Koba National Park in southern Senegal, the river Gambia flows through the national park, and right by the river, we established our beautiful field site. And when I first went there to explore the situation there, whether this was a suitable place to do research, I mean, there are baboons everywhere, everywhere. And I thought, great. And the first day we approached them, and we could approach them for about, say, 80 meters or something. They didn't run away. That all looked really good to me. Great, so we started. And then it turned out nobody has studied Guinea baboons because they're so hard to habituate. It took us full 3 years until they finally did not run away anymore. So basically, I mean, it was very good that I could use my seed money to establish the site there, because, I mean, the DFG, they would have just like, I'm gone crazy, I'm not producing any results, because we are just trying to, you know, convince the baboons that they should stay put and we could actually observe them. And these are some of the pioneers. We worked with really a great team, combined Senegalese field assistants and German students. Really very special. And during this time, when we were trying to habituate them, Annika, who was one of the pioneers, she worked at that time as a field assistant. She sat in a hide by a water place and just watched who was coming, what were the groups, what was the group size and group composition of the animals that arrived at this water site in the morning when they came down from the sleeping trees, and then they moved off to forage, and then in the evening when they came back from their foraging trips and drank some more before they went back to their sleeping trees. And what you found was that there were all sorts of things going on. So it could happen that you had several smaller groups coming in, and then they would walk off together as one big group. Or you could have one big group arriving, and then they split into smaller subgroups. And then there was also something what she called mingling. So you had small groups, and then they came together, and then small groups left also. So there was no aggression between these animals. It was very unlike what I knew from the Chakma baboons from southern Africa, who are really aggressive and are very cohesive as a group. And when they see another group, then all hell breaks loose, and the males start to wahoo and to beat up their women. And no, but they were sort of very easygoing and looked all very, very different. And then she also found that on average, the number of parties, so these small subgroups, she termed parties, or we called them parties, that the number of parties that arrived and the size of the parties respectively varied with daytime and season. So in the afternoon, the parties there were more parties and they were smaller in size. So it looked like bigger parties were coming in the morning. But this was also, the same pattern was found. So in the dry season, we had more parties that were smaller, and in the rainy season, they were, when food was abundant, they were running around in larger groups. So not so surprising, but so that was, we still didn't know who we were seeing. Well, eventually we decided, okay, we have to capture them. We have to put collars on them, and we have to mark some of these animals, because if you know, sometimes we saw 300 animals at a time, and it's just, they very much look alike, especially at the beginning. I mean, and I still think that the juveniles all look identical, but my students don't. They think that the juveniles also look very different now. So what we decided to do is, like, we have to capture them, and then we put radio collars on some of the males so we could actually track them, and more important, we put GPS collars on a number of animals at the same time. So here, then we measured them, so we got really good morphometric data and weight data. And what was really funny was that, so when the first student announced the first measurement of the testis size, Dietmar, he's a senior scientist, he said, no, that's wrong, you have to measure again, that's much too small. But the testes are really very, very small. They have tiny testes. So here is Matze, and Annika and Mustafa trying to locate the baboons in the morning. So, and then the kind of data we are getting looks like this. So basically, so the blue one is the river, and the red star is our field site. And then there are two animals depicted here, Kurt and Dao. And this is, they spent the night in the same sleeping tree or very close to each other. Error is about 10 meters. And then every 2 hours, we get a recording of where they are. And then we can calculate, so this is a pattern for 3 other animals that are not together all the time. And then from this kind of data, over 18 months, we could calculate how much time they actually spend together and when. And Annika's PhD project consisted of analyzing the spatial association patterns. Firstly, she used these GPS data, but secondly, she also used focal data. So she was actually, at that time, the animals were habituated, so she could follow them on foot and really see, okay, who is actually sitting near Kurt or Ndao or Musa or whoever. And so she had 14 males in her focal observations. And then she also looked at the quality of the interaction. This is kind of stuff that we're not yet getting via telemetry. So we don't know who's actually grunting to whom and who's friendly and who's aggressive, and so you know, you still have to follow them. And then she did a relatedness analysis using microsatellite markers. And if you want to read up on this, the paper just came out last week in PNAS, where most of the results that I'm talking about now are summarized. So what we did here is we plotted the association patterns from the GPS data. And so the animals that have a very high association index, like 1 up here, they're basically together all the time. And from the 83 diets that we had in our sample, most of the diets did not spend time together. A diet, oh, it's 2 animals. Yeah, you and I are a diet. Yeah, so it's not diet, not, yeah. Good. So 83 pairs of animals, and some of them spent really most of the time together within 100 meters, and many animals did not spend any time together, but then there was also this intermediate. So some animals spent about half of the time together. And then you can run an application, it's called change point analysis, to find significant breakpoints in the distribution of the data. And we found 2 breakpoints, and one said, okay, there's a difference in the difference between those that really don't spend time together and those that do. Basically here, the lower, it was really well supported, and here that is less well supported because, of course, here we have fewer diets in there. But still, it's a significant breakpoint. And we said, okay, this reflects what we actually also see when we're on the ground, that we have these small groups that we termed parties before. And then these parties, within the parties, or the parties may associate with other parties to form gangs. And then all the baboons in the area, so every now and then they do see each other and they cross paths, and this would be reflected then in the, say, 5% proximity that we get. So we call all the animals in the area community. Okay, so this is just from the GPS data, and it really fits nicely from the what we found when we looked at focal observation data. So just by running around and seeing, okay, this animal is sitting next to that animal, and this is sitting next to that animal, and so just by scoring who's actually sitting next to each other, we find exactly the same pattern here. This is the results from a cluster analysis. And for animals that spend a lot of time together, they are here, for instance, or these 3 spend, Peter and Usman spend really a lot of time together, but Anise also, or Hook, Sane, and Samuel spend a lot of time together. So these are parties, members of the same party. And then here you see these associations here very nicely. This is then at the gang level. And then, as you can also discern from this graph here, that these associations are relatively stable over time. So we find some of the same animals together that we were together in 2010 are still friends in 2011. And actually, I can tell you, some of them are still friends today. Some have died, but these are relatively long-term friendships or relationships between the males. But we see also that there are some dynamics. For instance, here in 2011, one animal joined this group of 3 males, Ba, and became a member of their clique or party. And then, of course, some animals get eaten. So we found one animal with just the collar was left, and the collar didn't look good. Looked like somebody had chewed on the collar quite a bit. So this is what happens when you do field research. And we see also some fluctuation. But overall, there is this pattern of consistent parties, and then parties predictably group up with other parties to form gangs, and then we have several gangs in the community. And what was really interesting is when we saw this association pattern, we thought, this looks like you really have brothers in these gangs, and they are more related to one another, or maybe cousins, and we also found that some actually looked quite similar to each other. And then we find a sort of slight signature of genetic relatedness, that male animals within a gang were indeed a little bit more related to one another than animals that were in different gangs, if you do these pairwise comparisons over and over again. But our notion that the parties consist of highly related animals was not supported. So there are highly related animals within a gang, but there are also highly related animals outside the gang. So if you look at, outside the party. So if you look at a gang and you compare the relatedness of members that are in the same party, they are not more or less related than the members towards the other party in the same gang. So it's not that you just have, a bunch of brothers or cousins that hang out together. It's a bit more complicated. And what is also striking is that the males do exchange quite a bit of affiliative, that is friendly behavior with one another. And they do that at twice the rate as they fight with one another. Agonism is nasty behavior, dominance behavior, stuff like that. And I had studied, as I said before, the Chacma baboons, and male Chacma baboons would not come near each other except to fight. And they would never, ever, ever exchange any friendly signals or behaviors with one another. They wouldn't touch each other, unless they're perhaps brothers and still very young. But once they're adult, forget it. And so that was really weird. And then they had one thing that I had never seen in Chacma baboons or any other baboon species for that matter, are these greetings. So the greetings are highly stylized, ritualized, weird behaviors that the male engage in. And it looks like this. So he's now strutting away. So there's a lot of penis pulling. So I say, they really have to trust each other. Hip touching, and then sometimes they do these head banging things. It's really bizarre behavior. So there are different ideas why they're doing this. As you can see, they're highly aroused when they do it. And there are two not mutually exclusive ideas what is going on. So it may serve the bond testing. So actually, males that are friends do this to one another because, if you want, somebody else lets you touch your crown jewels, you have to trust each other. And so it could be, one function. It could also be that it serves to avoid agonism. So there is actually some tension in the group, but they don't start to fight. Instead, they do greeting. And this way, they eventually are able to calm down. And we are now doing a very detailed behavior of the analysis of the sequences of different behavior patterns to actually address this in the wild. What was also striking was that in none of the aspects that we looked at, so proximity, relatedness, affiliation, greeting, and agonism. Was there any correlation in these very small networks, I have to say. So here, you see the proximity data. So here would be from, this is 2011. So we have here the parties, 2 parties in one gang and 2 parties in another gang. And then we found in each party 1 highly related dyad of 2 males, but the others weren't related at all. And so now, of course, this led us to formulate the conjecture that perhaps dyads establish themselves along, really, that you have 2 brothers or cousins, and they grow up together, and then other males join them. But we need long-term data to address this. Then affiliation is mainly within the dyad, but it's not along the lines of relatedness. So it's not just that the brothers are friendly to one another. Also, unrelated males can have very positive affiliative bonds. Greetings, that you can also see there is some variation between gangs, mainly within the party, and agonism is across party. But there's no correlation whatsoever. So one very prominent finding in many types of behaviors in social groups is that kinship is important. And in this case, kinship does not predict the relationship quality, neither in the positive nor in the negative sense. And what's also striking is that we were unable to discern a linear rank hierarchy. So if you study Chacma baboons, and even if you're not a very experienced researcher, it takes you about a week, and then you know who's the dominant male and the second and third, fourth, and so on. Very simple. It's very clear who's the boss and who's not. In these guys, you know, even after now 4 years of systematic data collection, we can say, okay, there are some males that are more important than others, but we are unable to say what the rank hierarchy really is, if there exists a rank hierarchy. And that's actually one very interesting discussion I'm having with my colleagues. Some say, you just need more data and then you will discern the rank hierarchy. And I say, I'm pretty sure now that we will not be able to do it, because most of the males just never ever exchange these dyadic patterns that allow you to infer who's dominant and who's subdominant. They just don't do it. And what's also striking, and this fits to the pattern that I mentioned earlier from our early observations, is that, so these are the home ranges of 3 gangs in our area, and they basically use the exact same home range. I mean, there's like 90% home range overlap. So there's no territoriality at all. And also, as I mentioned before, when gangs meet each other or parties meet each other, it's like, you know, it's like you're in a mall. It's like, oh yeah, hi, there are other baboons, you know, okay, let's move on. So they really don't care. And this is very different, again, from the southern baboons. So in sum, we found evidence for a multi-level system, we think. So it looks superficially very much like what we see in the Hamadryas. So we have the parties, and then the parties team up to gangs, and gangs can team up to the community. Now the question, of course, always is like, you know, what we see, does this correspond to what the baboons see and what the baboons care about? And to get a handle on this, like what do they think is important, what is important grouping? One thing you can do is to do field experiments, and one very famous technique is to do a playback experiment. So this is a Nagra speaker somewhere hidden in a way that the baboon wouldn't be able to see it, and then you play the vocalizations. And what we did is we played vocalizations from animals in, we call them the neighbors. They aren't really the neighbors, but they're using the same home range, but they're in a different gang. So from a different gang in the same home range, and then from complete strangers, 50 kilometers away, they've probably never heard them before, and then as a control also from males of their own gang. And we were interested, I mean, we conceived these experiments very early when we still didn't have a full understanding of what the social organization was really going to be like, but we suspected, well, maybe it tells us something about whether they have learned to ignore the neighbors because they know they don't pose a threat, whereas they would respond strongly to strangers. So that was sort of one of the expectations we had formulated. And then we did these experiments, and this is one of the videos. This is a PhD project from Peter Machi. Now let's see. Okay, this is a male, and he hears grunts, and I'm going to put the microphone here. He hears grunts from another male, and then you can see whether you can see his response. He almost tiptoes over, yeah. So he's very interested. It's like, what is this? You know, what is going on? And I thought, wow, nice, strong response. Probably in response to a stranger. No, they don't care about strangers. They couldn't care less, and they also don't care about whether there is a male from another gang in the area. They really only care about their own males in their own gang. And they try to figure out what's going on. You know, who's interacting with whom here? You know, who is friendly to whom? I need to check out. This is really the sort of consistent, strong responses where only two grunts from vocalizations indicating some interaction between a male and their own group. And I have to say, these were males that we played back from the other party in the same gang. And I think that is sort of explains this pattern fairly well, because they want to know what is going on in the other party, because they are highly relevant in terms of maybe low-level competition. So I think that sort of explains the response really. Now, I've been talking about males all the time, and there has been, you know, a lot of criticism in the field of primatology because all the early studies in the 1960s and 70s, they only focused on males and males' interests and male strategies, and for males, females are a commodity, if you will, you know, and then some feminist primatologists said, you know, what are you doing? I mean, you cannot just look at the males and explain the society. You really have to look at the females as well. So what are the female strategies? And we did the same thing. We started with the males. They're bigger, they're more different, they're not as shy, so we started with the males. But now, of course, we're also looking at what the females are doing. Or do females fit in? And this is a social network analysis. This is the 1 meter proximity data for males, and males are discerned in squares and females in circles. And these are two different parties in one gang. This is a PhD project from Adelia Goff. And what you can see is that each female is affiliated with one big male. There are also some secondary males that are affiliated with that same big male. But the typical pattern is that each female is mainly, you know, has one tie with one big male. And the males, in turn, they may have something between 1 and 5 ties. We also have one male that was able to have 6 females at the same time. But so the number of females varies quite substantially between the different males. So this is really, it looks very similar if you just look at the male-female relationship to what was found originally in the Hamadryas baboons. But there are also some differences. So in Hamadryas baboons, it's basically like this. A young male will try to acquire 2 or 3 females that are also young when he's still very young. And then he will hurt them consistently and make sure by neck biting, you know, that they do not walk away from him. If they like go further than 3 meters, they will be beaten up. So it's a very, very strict regimen and a lot of male coercion and enforcement and policing. And in the Guinea baboons, this is really different. So here's a little scene, just to give you sort of a flavor of how dynamic the system is. So again, this is a different gang now. But again, we have males in squares and females in circles, and please pay attention, especially to Annika. So it's October, everything is good, you know, it's been like that for a while. And then November starts, and Annika decides to leave Peter and join Dur, where Fantu leaves, joins Ba, then Daphne leaves, and then Mimi leaves Ndur, and then Annika has had it and goes back to Peter. And then she stays put for 3 weeks, and in December she actually moves to Usman. So now Ndur has lost all his females, Ba has lost 1 female, yeah? So there was this whole thing, and we've seen this now 2 or 3 times, that one female starts to change, and then this sets into motion this entire chain reaction of switches. And we are not able so far to predict which female will move and when. I mean, it's basically overnight, everything is fine, just you do your focal observation one day and she is with Peter, and then the next morning she is with this other male. And you have no idea what is going on. And it doesn't seem like the male is trying to get her back or anything. They're not fighting over the females. It really looks like it's the female say. So the male may make some attempts that, like when he sees she's sort of walking away, to do something about it, but he's not very successful. And as you've seen, also some females do not only switch within the party or within the gang between parties, but they also leave the gang. And so this is also in line with the view that females are actually dispersing here and responsible for gene flow. So in quantitative terms, we've observed 14 females now. Each female associated over the entire time with anything between 1 or 6 males. So Annika, I think, was the one with the 6 males. The mean association time is something, 270 days. Range is between 15 days and 507 days. So this is so far, what the kind of data we have collected during this one specific project. So there's quite some variation, and of course now we are asking, like, what are the causes? What is also interesting, despite the fact that males are not really policing the females and hurting them, females, once they've chosen a male, they stay with him. And almost all of the observed copulations were with that primary male that the female chose to be with. So in this sense, once the female is with the male can be sort of sure that she will not go behind the tree and mate with somebody else. So he doesn't need really to fight over her at in this point. So this gives rise to the question, like, what do females want? Why do have some males 1 female, and why do others have 6 females? And what makes some males more popular than others? And one is, it could be that they prefer nice males. For instance, here, male is sharing meat, actually, so that's something that's unheard of among monkeys. It's been discussed highly in chimpanzees, but the Guinea baboons also share meat. So he tolerates that she takes meat, but that would also not happen in Chacma, for instance. Or do females prefer males with strong bonds? So perhaps that if you are actually choosing a male that has 2 really good friends, then you feel safer against bachelor threat or something like that. So these are ideas we're discussing. We do believe that female choice plays a big role in the species, and the males, they have these great coats, and perhaps that's also an indicator of male quality, and female may make their choices also based on these kinds of indicators. But we have to look into this. And then, of course, there's also the question of female competition. So perhaps the resident females may also play a role, whether a female that comes into that 1 male unit will actually be successful and be able to stay, or she comes in and will then expunge another female. So these are all really interesting questions, but we need about at least 10 years data to say something what is going on. What is also striking and very different from the Hamadryas baboons is a point that I didn't highlight before, is that the females maintain relationships outside their units, outside their 1 male units. In the Hamadryas, that wouldn't be possible. If a Hamadryas female grooms another Hamadryas female that belongs to another male, she will be beaten up. So here, no problem. So what we think what is very special about the Guinea baboons is that they have these strong male bonds and something what we call for now female freedom. So females actually do have a say with whom they affiliate. Now, you had to believe me all the time when I was saying, well, that they're different from the Chacma baboons, they're different from the Chacma baboons. But of course, it's nice to step back and put some data to it. So actually, one of my more recent PhD students, Urs Kalbitzer, he did a completely comparable study in the sense that he went back to Botswana, to the same group that I had studied years and years before, and collected data there, and then used the exact same protocol to collect data from the Guinea baboons. And we need much more data of this kind. It's very nice to do meta-analysis, and I often hear people saying, yeah, in meta-analysis, this is the way to go. It would be so cool to have meta-analysis. But if you really nail it down and look at what people are collecting very different types of behaviors, and it's not simple or really, frankly, mostly impossible to pin it down and really discern, you know, from the raw data, really good behavior rates. People have different definitions, and I think one of the big challenges for the future for this kind of research is that we develop standards and that we have publicly available databases where we adhere, you know, to the similar protocol, the same protocol, and then we can actually compare our data at this. And Ur's PhD project is like one small step into that direction. So he actually calculated behavior rates. So here's for agonistic bouts, dark gray are the Chacma baboons, and white are the Guinea baboons. And you see agonistic bouts, so yeah, occurrences of aggression. And then supplants are sort of a more subtle way of settling who is actually superior to someone else. It simply works like this. So the higher ranking animal approaches the lower ranking animal, and the lower ranking animal has to move away. And this is very nice. It's sort of subtle. They don't have to fight about it, but sort of this just ensuring, you know, to make sure. And if the higher ranking animal wants to interact with the lower ranking animal, she has to signal that she's not going to do anything if that female doesn't walk away. So it's all very, it's like Denver Clan, you know, like, or Dynasty, watching Dynasty just in real life. So these data clearly show that male Chacma baboons are much more aggressive against other males than Guinea baboons, and they're also more aggressive against females than Guinea baboon males are against females. And Guinea baboons don't really have supplants. I mean, very low rates of supplants. There's a big advantage of being a Guinea baboon. You're generally less stressed out. So also looked at the physiological cost of being a Chacma baboon, higher, and then in relation to rank. Now, of course, with the Guinea baboons, this analysis is somewhat, yeah, you have to treat it with caution because we don't really have ranks. Yeah, so, but even if you permute the data, no matter how in which order you put the males, there is no correlation with stress hormones. Whereas in the Chacma baboons, it's relatively clear that the high-ranking males are much more stressed out than the low-ranking males. So they're constantly threatened to be toppled, and then that, you know, so they have to handle all sorts of situations all the time. That seems to be stressful. And then we were interested in elucidating the genetic foundations, and this is just a very small snippet here. This is parallel research is going on in humans. Can we find any genes that are associated with higher propensity of aggression, impulsivity, not being able to, you know, control your temper? And a very prominent example is the monoamine oxidase A, an enzyme that catalyzes neurotransmitters in the brain hormones, if you will. And there are two, in humans, there are two forms, a long form and a short form. And there is an intriguing gene-environment interaction. So when you have the long form, no matter how shitty the world is you live in, you tend to be a nice person. Yeah? But if you have the short form and you are in a bad environment, then this raises the propensity of being aggressive in certain situations. So this is contested research. Yeah, there's always, you know, you have to sort of take it with a pinch of salt, but that is one pattern that has been reported for humans. And now we were interested to see whether, because in the Guinea baboons we would say, okay, these guys should all have sort of the equivalent of the long form because they're just so laid back, the hippie baboons. And maybe the Chacmas all have the short form and they don't even need a bad environment, they're just always aggressive. So we did not find exactly what we predicted, and also baboons, they do have also monoamine oxidase A, they also have these linked polymorphic regions, so they also have short and long versions, if you will, but they're different from what goes on in humans. And we don't even know in which way they're affecting, for instance, transcription rates. So how frequently is this gene actually transcribed and sort of has a biological meaning. We don't know this yet, but we do find an intriguing geographic pattern, and the next step is to see whether there is actually selection for the PAP-A, so for the shorter allele in the more aggressive species, but we don't know this yet. Now, of course, sort of to summarize, or to come to the end, more or less, what are the causes for this variation between different species? And one classical idea is it has something to do with the ecology. Do you live in a land of plenty? Then you can be a nice person. But if we compare the habitats the different species live in, that doesn't provide the full explanation. The same holds for predation pressure. If we map predation pressure onto different baboon species, that's not the explanation why they are so different. And one very prominent theory at the moment is that we have to go back in evolutionary time and look at the evolutionary dynamics during the range expansion. So baboons originated in southern Africa in about 2.5 million years ago. And then they started spreading across the continent until they met the impenetrable rainforest. And then there were these interglacial periods when the climate became cooler and then corridors opened up. But before that happened, we had a situation where they basically sort of hit the wall. They couldn't progress any further. And in this very specific condition, at the fringe of the range expansion, dispersal is detrimental. You don't want to walk into that forest and never come back. Never find another baboon you can mate with. You want to stay at home. So in this situation, it's actually there's a positive selection specifically if the population density is very high, that you just become a homebody and you stay at home. And this eventually leads to an accumulation of males that are related with one another. And if you have more related males, they tend to become more cooperative via kin selection because they are brothers and cousins, and then you start to become nice to other males that are in your same group. And perhaps this sort of, at the very fringe of the expansion, promoted the emergence of male tolerance and cooperation. And then these corridors opened. And then there is a phenomenon, it's called gene surfing, that if you have a population and at the fringe, the population genetic structure is somewhat different, you get the subselection there, and some genes that have been selected or that could also be random, that are occurring at this outer fringe of the distribution, now become the source of this new gene pool, basically. So you can have a rapid change in the population genetic composition at the fringe of the distribution. And not just something like genetic drift. You can really have these nonlinear phenomena in terms of the genetic composition. And these two, basically, we are sort of the founders for both the Hamadryas baboon and the Guinea baboon. So that's the story. We don't know if it's true, but it sounds good. But what we do know to support this idea that spatial dynamics play a role is not only studies that looked at the emergence of cooperation, but also the maintenance. And this is work, among others, by Katrin Fehl and Dirk Semmann, published in Ecology Letters, and of course, the massive amount of work by Martin Nowak. And if you play public goods games where people give an endowment and then depending on their behavior, they get money back or not, and you start a situation where everybody is linked to 3 other people, and then they do this cooperation game, and depending on their experience, they are able to choose not to play with a certain person again. Then you will see that these clusters of cooperators emerge. So the population that is initially sort of even, all of a sudden you have situations where you have these subgroups of cooperative and subgroups of not cooperative people together. So spatial dynamics play a much bigger role, and I think we're becoming increasingly aware at very different levels how important space is in evolution, not just time. And so once you have such a situation, it's like a positive feedback loop. So if you have cooperators together, they tend to cluster in space, and when they're clustering in space, this maintains their own cooperative tendencies. And now for something completely different for the last 3 minutes. Now, my own background was really trying to understand the link between social behavior, social organization, social structure, and intelligence, and signaling behavior, and test the assumption that they may, give rise to one another, may be linked to one another. But if you study baboons, you say goodbye to this hypothesis relatively quickly. So I've told you a lot about the difference between the Chacma baboons and the Guinea baboons. But if you look at the signaling behavior, and these are spectrograms, these are like the music score, the musical sheet of their vocalizations. So you have the frequency on the y-axis and time on the x-axis, and here are three different call times discerned, and there is hardly any difference. I mean, the Guinea baboons sound a little bit higher. I can see if I can play this. So that's a Chacma baboon wahoo. It sounds quite impressive. Not so impressive. And then the grunt. And this is a Chacma. So all in all, most of the differences can really be explained by size differences. The Chacma baboons are substantially larger, and if you control for body size, the differences disappear, except for in the early one, this one, the wahoo. This is the display signal of the Chacma baboons, and it's really exaggerated. And the Guinea baboons don't use it as a display signal. They just make these calls when they see a lion or when they've lost the others, and so that's a big difference also. So as far as signal evolution is concerned, across all the baboon species that I looked at, the vocal communication and also the other behavior patterns. So I asked all baboon researchers that have been studied them in the wild to send me the catalog of their behaviors, and they're virtually identical. I mean, the greeting is something that's derived, the male greeting, but the simple like threats, affiliative gestures, all the basic stuff has not changed at all. And I think this goes back to the fact that most signals have two functions. The one is come here or go away. And this basic function, is retained in the different species. So there's no pressure here to evolve a larger diversity. And there is a big hypothesis out that social and vocal complexity go hand in hand. And as far as for the Guinea baboon, for the baboons, I can say we can refute this hypothesis. It's not always the case. So I want to send you home or to dinner, and hopefully some beer or wine or whatever. So there's 3 key messages. There is no such thing as the baboon. Yeah, whenever you read a paper, something about baboons do this and that, you have to say, no. Which baboons? What do they do? So there is a huge variation among baboons, and there is even greater variation among all other primate species. I mean, we tend to talk about monkeys, Affen in German, and Affen sind nicht Affen. Yeah? There are really, really, really big differences between the different species in terms of numerous features, and that's what makes them so interesting. How can we explain this astounding diversity in primates? Then spatial dynamics play an important role in social evolution, probably not only in social evolution, but certainly in social evolution. And signal evolution may be astoundingly conserved. And this is, of course, relevant when we try to understand the evolution of language. How can you have such a highly conserved system? That's also true for chimps, highly conserved. There's no variation, well, small dialects, but really, all chimps use the same sounds. And then what happened? That at some point in time, somebody started to talk. And the outlook, grim. Our field site is in West Africa. I'm really, really worried. I don't know if we are able to answer any of the questions that are raised that I thought are so interesting. We have no idea when we will have to pull out all the researchers and what will happen. It's a complete catastrophe, and I have no idea about the inertness of the politicians on earth. I mean, yesterday at least Barack Obama pledged something, but when I read the German newspapers, I just want to scream all the time. I mean, it makes me crazy that so little is happening. I mean, Médecins Sans Frontières, they started in April to say, this is a catastrophe. It's gotten out of hand. We can't control it. And nothing happened. And I don't understand it. And this is something that's more important than Guinea baboon greetings. If there is a way to contain this, if we are able, I don't know, to proceed with this research, this is what we would like to look at: the social dynamics, the development of bonds, female choices. We want to know more about what they know about each other. And then we want to start these broader comparative analysis, including behavior, ecology, and also, of course, omic data, so genetics, transcription data, physiology, etc. I have numerous people to thank, specifically our Senegalese partners. It's great to work with them. Once they accepted that there can be such a thing as a female director, it worked out fine. On the ground with the field assistants, it's really great. It's a wonderful camp, a wonderful group of people. And then, of course, my wonderful students that really do all the real work. And I thank you for your patience. Thank you.